Very good evening to all of you. A warm welcome to one and all. It's a very happy occasion today for us because we have with us Professor Raj Mutharasan from Atlanta. He's a retired professor, and a formal introduction will be given by Mr. R. Balakrishnan, the honorary consultant to the Indus Research Centre of the Roja Muthiah Research Library. And this lecture is organized under the Indus Research Centre as part of Professor ANandaKrishnan Endowment Lecture Series. Professor AnandaKrishnan, many of you will know that he was an educationalist. He has served as vice chancellors for a few universities. He was the chairman of IIT Kanpur, and he was also the trustee of the Roja Muthiah Research Library till he passed away, unfortunately during COVID times. I fondly remember him, his contributions. While he was a trustee, in one of the meetings when we asked, we were deliberating about starting an endowment lecture series. He said that he will contribute, and with that contribution, we started this endowment lecture series. That's incidentally the first endowment lecture series that we started. And now we have several endowment lectures going on. And now Mr. R. Balakrishnan will be will be formally introducing the speaker. And I also would like to welcome Mrs. Raj Mutharasan. So it's so kind of to come with him. Thank you, ma'am. Over to Mr. Balakrishnan.
Good evening, everybody. I really consider this to be a privilege to welcome and introduce Professor Raj Mutharasan, whom we know for quite some time and had an opportunity only during last year to meet him in person. Today is going to speak about the exploring Indian and Tamil ancestry through genetics, methods and recent findings. Professor Raj Mutharasan, an accomplished researcher and educator in the field of chemical and biochemical engineering. Professor Mutharasan earned his bachelor degree from IIT Madras, India, and his PhD from Drexel University in 1973 in US. Over his distinguished career, he has guided 30 PhD dissertations and over 40 MS theses, Master of Science theses, contributing significantly to the advancement of the field. His research spans process control, biochemical engineering, process metallurgy, and more recently, biosensors, with funding from prestigious agencies like NSF, NIH, and USDA. I hope that such funds are going to continue and exist, among others. His pioneering work in biosensors has led to 12 patents. He has got 12 patents in his through his studies, primarily focused on his invention of the cantilever biosensors for DNA, cancers biomarkers, and pathogen detection. Of significance is methodology for measuring a few hundred cancer biomarker molecules. Professor Mutharasan is a fellow of AIChE, must be all American Chemical Engineering Association, AAAS, AIMBE, and has served in editorial boards of prominent scientific journals, including Metallurgical and Materials Transaction, biosensors letters. His contributions have earned his notable honors, including Drexel University's Research Award and the Distinguished Service Award, as well as endowed Frank A. Fletcher Professorship. In addition to academia, he has served as Interim Dean of Drexel's College of Engineering, as a Program Director at the National Science Foundation, Washington, D.C. He was honored with the Distinguished Alumni Award by IIT Madras and has provided his expertise to organizations such as ALCOA, Schering Plough, BioChem Technology, Kline, and the United Nations Development Program, UNDP. This is formal introduction. I would like to get slightly informal for maybe one more minute. I would like to welcome Mrs. Raj Mutharasan, Karpagam Raj Mutharasan. We had an opportunity. Last year, நாங்கள் அவங்க வீட்டுக்கு சாப்பிட போயிருந்தோம். அட்லாண்டாவில். ஒரு அருமையான சூழல், மிக அருமையான புகைப்படங்கள். அவர் வந்து முத்தரசன் வந்து ஒரு சிறந்த புகைப்பட கலைஞரும் கூட. தமிழ்நாட்டில் நம்ம பார்த்துருக்காத கோயில்கள், சிற்பங்கள் இதையெல்லாம் காட்சிப்படுத்தி இருக்கிற விதம் வந்து அநேகமாக ட்ரான்ஸ்கி மருது அடுத்தோட அமெரிக்காவுக்கு போகும்போது அவர் வீட்டுக்கு போனீங்கன்னா அந்த படங்களை வைத்திருக்கிற முறையை நீங்கள் கட்டாயம் விரும்புவீர்கள். அப்படிப்பட்ட ஒரு அதை பார்க்கும்போதே உங்கள் நினைவு எனக்கு வந்தது. ஒரு அழகியலாக காட்சிப்படுத்திருக்க வேண்டிய விதம். அவர்கள் எங்களை உபசரித்து எங்களுக்கு மதிய உணவும் கொடுத்து விட்டு, மதிய உணவு கொடுத்து இந்த தாம்பூலம் கொடுக்கறது மாதிரி சுந்தர்கிட்ட வந்து ஒரு 10 லட்சம் ரூபாய்க்கு நூலகத்துக்காக ஒரு செக்கையும் கொடுத்தாங்க. அது தட் வாஸ் தி ஐசிங் இன் தி கேக். அவர் எங்களுக்கு தெரியாது, எங்களுக்கு மதிய உணவு கலைச்சிருந்தாங்க. மதிய உணவுக்கு போயிருந்தோம். நாங்கள் இந்த நூலகத்துக்கு ஆதரவு திரட்டுறதுக்கு தான் போயிருக்கோன்னு தெரியும். அந்த மதிய உணவையும் கொடுத்து ஒரு 10 லட்சம் ரூபாய்க்கு செக்கையும் சுந்தர்கிட்ட கொடுத்தாரு. இவ திரு முத்தரசன் அவர்களையும் மிஸ்ஸஸ் கற்பக முத்தரசன் அவர்களையும் சென்னைக்கும் இந்த நிகழ்ச்சிக்கும் வரவேற்பதில் நான் மிகவும் மகிழ்ச்சி அடைகிறேன். இங்கு வருவதற்கு முன்னால் அவர் வந்து முழுக்க வட இந்தியாவில், குறிப்பாக பீகார், உத்தரப்பிரதேச பகுதிகளில் மௌரிய ஆட்சி தொடர்பான, மகத பேரரசு தொடர்பான பல நினைவு சின்னங்கள், அகழாய்வு தடங்கள்லாம் போய் புகைப்படங்கள் எடுத்துக்கொண்டிருந்தார். அது அடுத்த முறை அமெரிக்கா போகும்போது அவர் வீட்டில் அந்த படங்கள் இடம்பெறும் என்பதை நான் அறிவேன். உங்கள் சார்பாக அவரை வரவேற்பதில் மிகவும் மகிழ்ச்சி அடைகிறேன். தேங்க்ஸ் லாட். வெல்கம் சார்.
Before we hand over the mic to Professor, I would like to give a small memento to Professor. May I request our senior artist, Mr. Marudu, to kindly, Trotsky Marudu. Please.
Thanks. Reporter.
Thank you, sir.
அந்த புத்தகம் இப்பதான் ஒரு 2 மாசத்துக்கு முன்னாடி வாங்கினேன். பரவாயில்ல, 2வது காப்பி. Thank you very much, Balakrishna and Dr. Sundar Ganesan. Thank you for a very nice, warm welcome. I'm glad to see the room full, almost no empty chairs. So that's good. That's a good news, right? The key thing that I wanted to share with you is some of you are curious about this area, and some of you may have heard about it through various different channels. I was invited by a study group to give a talk similar to this in November of last year, and that apparently were excited a lot of people. There were a lot of questions, so there was a follow-up session. So what I'm going to present today is a sort of a summarized version of that. The space is very large. My goal is to give you a sense of how this particular field has evolved and important things to remember. That way you can go forward learning a little bit more. And it is not out of the scope of almost all of you. So the key thing to remember is take this as a learning session rather than getting an opinion from any particular expert view, if you will. So my first term is disclaimer. I'm not an expert in genetics, but I've dabbled in genetics about 25 years ago, trying to put in certain genes in the hybridoma so they will express a particular protein that's of pharmaceutical value. And of course, I found that to be not so interesting because it's a very tedious affair. And out of 100 trials, you may get 1 success. So I moved on further into that because during that problem, I had to figure out whether the cell was doing what it is supposed to do. So I had to make a measurement system that way I can figure out early detection of the expression of a particular gene. And so I moved on to sensing technology. That's where I spent the last 25 years. And of course, that included DNA and also biomarkers, particularly for cancer, also toxins and a whole series of other things. So I'm not in the core space of genetics, but I'm very familiar with this space. And the second thing to remember is that I followed this area because simply out of research interest. since about 2000 fairly closely, and most of you will be able to read these articles and understand the main ideas. You may not understand the details of the methodology, but you'll be able to understand the conclusions and the arguments they make. And so I encourage you to read in this space. Fortunately, many of the research papers are in open access, and therefore it'll be available to you. And I will write through my slides. I'll have at the bottom the source of that information I'm sharing with you, and they will be, most of them will be available except one or two, and so you can dig into the details. And most of you are interested in where you came from, and I think Tony Joseph has done a nice job of publishing his work, which is a derivative of David Reich's work, which came out almost a year earlier. And we have obtained a great deal of rich information in the last 7 years. So the field is really about 7 years old, although the fundamentals of this. Hmm? This is better. Either. Small technical glitch, I suppose. Okay. So what I'm going to present today is the research results of leading scholars in the field. So I'm used to giving talks on my own research and things, new knowledge that my lab has generated. So this is a time when I'm giving a summary of other people's research. And so keep that in mind. So I may not know all the full details, but I understand the basic conceptual framework and which I wish to share with you. And the real issue started somewhere in the mid 1990s. National Geographic magazine had a major article on Out of Africa program, which funded a whole series of projects headed by Stephen Spencer Wells. And of course, from the Indian side, Professor Pichayappan was the participant on the Indian side. And of course, I'll share some of those results too. The key thing to remember is the methods and approaches are evolving, and there's a lot of publications that have come out in the last 2-3 years. So it's an enormous rich literature. And I always caution my students, what is true today may not be true tomorrow. That's typically what a scientist says. Remember, we believed Newton was accurate until 1908, until it was proven he was wrong. Not wrong, he was approximate. So body of knowledge in this space has the same tenor. So what we share with you is true as of now. It may not be true going forward because new information and new data and new methodologies will come into play and will refine the body of knowledge that currently exists. So keep that at the back of your mind. And this is sort of my rough outline. I'm going to give you a background, a little kindergarten story of molecular biology, of genetics, basic elementary genetics, which is related to ancestry. And then I'll take two examples arbitrarily chosen to illustrate how genetics can provide light on history. That is, genetics is really part of history now in the modern concept, although that it is a separate field at the moment. So I'll share two examples from that because that'll gain interest on your part. And then I'll take recent research results, approximately 3 or 4 papers, and share with you because it has meaningful application in the case of Indus Valley and of course India and of course Tamil Nadu. And then finally, a few key results from ANSI, which is Anthropological Society of India, which is based in Calcutta. I wasn't aware of it until not too long ago. They published a major paper of making very large genomic studies of Indian population. I should caution you here. All of the studies of the Indian population done by all researchers, no exceptions in my knowledge, have done it based on caste or religion or some division that exists in society. And none of them, from what I know, have made random sampling. They all picked whoever they came in front and they took. So the data, when I look at the data, there's a great deal of variance, and that's because of their sampling method. So the data I'm going to share with you has got a lot of what I would call as errors, but the trend will be the same, but the errors will be there. And that's the nature of this particular field. And for whatever reason, all our Indian scientists have chosen the population to look at their genetics based on divisions in the community. And so we get what I call as a scientist, a biased data, not a what I would call a randomized data, not representative data. But that's the nature of this field. We have to live with this. Even ANSI, the Anthropological Society of India, did the same thing, even though they have several leading scholars in the field. I feel sad when I see that when the data is not representative, that means you can't draw general conclusions. You can only draw partial conclusions. Okay, and then finally, I'm going to share with you the real world example because I was curious about this. So I've done my own DNA analysis over years, and I've participated in the National Geographic Magazine project, and subsequently we're using a commercial enterprise about a few years ago. So I'm going to share with you to validate some of the findings people have found because my data appears to provide support for some of the arguments that Balakrishnan AI has made in terms of Indus to Valley argument and many others, very other similar arguments. Okay, let me get started. Now, the data on ancient genome, that is people who lived 4,000 years ago, is really recent phenomena. Only 7 years ago, it started taking up. So this number, you'll notice that about 2017, it begins taking off. And now we are over 10,000 genomes of ancient people. People who lived 2,000 years ago, 4,000 years ago, 5,000 years ago have become available. Bulk of them are in Europe and Russia. The climate preserves it. And Central Asia, Africa, and Oceania, all of this is only 500. In India, how many? 1. This was the Iraqi Gargi genome. And of course, we have some Putnam, Kerala, that was partially, they didn't do a Y haplogroup because they weren't able to extract properly. They got partial information. And then we have some Himachal Pradesh in a high altitude lake, but that is not really as representative of India. So I'm discounting those two studies. So we have only one genome to represent 1.4 billion people. On the other hand, we have about almost 10,000 to represent less than 500 million people, right? So we're not participating. Mr. Modi has to get on his butt and do something to make this accurate. Well, we have a climate, warm climate deteriorates the DNA material, and therefore you cannot recover that easily. And people have had difficulty with that too. So there is also a situation with that. But I've heard from Spencer Wells directly at one of the conferences that we have good preserved ones at Egmore Museum, but they haven't been approved for analysis. But that might change because there are more labs in India that are beginning to adopt technologies that were developed primarily at Harvard and Max Planck Institute to do ancient genomes analysis, if you will. Hopefully that'll change. So the story we're going to hear about is really based upon most of the rest of the world's genome, ancient genome, and a very tiny bit of Indian genome. But even then, it has provided us some rich data, and there are a lot of estimates that we have to live with. Okay, kindergarten story. There are two markers that are considered important in trying to figure out your ancestry. All of us have 23 pairs of chromosomes, that's 46. And out of that, one of them is a so-called sex chromosome, X and Y. Y is present only in a man, not in a woman. And Y chromosome goes through mutations periodically. And the mutation can be measured quite readily, quite easy. You just buy a kit from a company and then you run through your PCR machine. Within a day, you'll be able to get that result. It's a very standard protocol, if you will, almost like titration. Some of you might remember from your high school chemistry class. And that remains the same between your father and your grandfather and great grandfather and all. But it does go through mutations in the orders of tens of thousands of years, not every 1,000 years. The second one is a mitochondrial DNA, which comes to you by your mother. And that goes through a greater level of mutations, and we can track those mutations too. And so your mitochondrial DNA will be the same as your mother's, and what she receives will be the same as her mother's, and so on and so forth. So you can think about this as a maternal ancestry and paternal ancestry, and therefore you can track your ancestry for you just making these two measurements. They're quite readily measured, by the way. So there are labs in India, I'm sure, that will be able to do this. I know CCMB at Hyderabad will do this. I do not know whether they're open to. Madhura Kamaraj University has a laboratory in this space too. And so also in Lucknow, there is another lab. There might be many other commercial labs. Now, you have each of you approximately has about 37 trillion cells, and they express approximately 20,000 genes. 20,000 genes are the intermediaries that create what are called as proteins or hormones, a whole series of chemicals that does the work for you. So in essence, you're a biological machine which is dictated by your genome. And of course, there are many other aspects to it which we don't quite understand. The key thing to remember is out of this huge genome chromosome, we're using just 1% or less than that for us to function. We do not yet know what the rest of it is really do. We discover them as things go on. One example I'll cite with you is Bristol Myers announced, this was about 15 years ago, that we had figured out how to stop cancer cells from coming out of the membrane. And they marketed a very powerful drug, supposedly. And then when they were administered to the patient, the human body discovered an alternate method of getting these cells out. So we learn things as we go forward. And of course, Bristol Myers took that drug out of the market because it was not effective. It was initially marketed as an anti-metastasis drug, but it was not successful. So we are still learning about what everything in our own genome does. And there are all kinds of theories behind it. We won't get into that. So the bottom line here is the Y chromosome and the mitochondrial DNA and their characterization carries the message of your ancestry. Now, the mutation occurs in the nucleotide. Nucleotide is a part of the molecule, if you will, of the genome. We don't need to get into. I don't want to get too technical because some of you have the population, I guess, here is very broad. These are the main markers of ancestry. Now, they're not the only two because you have 20,000 other genes, and therefore those genes also contribute to your ancestry. And for you to dig at them, the analysis is very expensive because that's a huge molecule. You got to sort all of that out. Most people don't do that type of analysis, and therefore it becomes a little difficult task. And so when we will, this body of knowledge for Indians has remained more or less at the ancestry part, Y and mitochondrial DNA, and less on the genomic part. But I think that'll evolve as future time it goes forward. Now, the converse is not true is important. The mutations are labeled A, B, C, D, etc. If you don't have a particular ancestry, does it mean you did not come from that particular lineage? That's not true. The reason is your Y could be hidden, but your genes will still be present. I have an example if there's a question about this, but I don't want to get into that detail at the moment. Any one question that I could answer at the moment? This is a typical professor talking.
Yeah, I saw some skeletons there, and you said about the climate and all that. So, will it be difficult to extract the DNA from such skull or bones?
I must preface that by saying I have never done ancient DNA extraction, although I've dealt with DNA. So experts in India say that is difficult to do. Apparently, Dr. Tangaraj from CCMB has said that. But remember, David Reich's lab has extracted DNA from Denisovans of 35,000 years ago. So I do not know whether the technique is inadequate or whether there were other issues involved. I think that's an area of strong activity required, because my belief, the typical one that they did in Harvard lab is take the molar because it's deep. If any of you have had root canal on a molar, you know this. It's very deep. The dentists charge you more for molars than other teeth, and it's well preserved. It's far away from the exterior. I believe there might be opportunities, but I must also point out with the Rakigargi, the extraction they did at Lucknow, they did 50 trials out of which one was successful, and they didn't get the whole genome. The number of SNPs they characterized was about 57,000. I'm remembering from reading some time ago. Normally, the number of SNPs will be in the orders of hundreds of thousands, 100, 200,000. So they got only part of the genome, not the entire genome. So there could be some difficulties. If the same sample was done at Harvard, I do not know whether they would have done better. So I think that's evolving technique, but I think that could be acquired. Okay. Only one question, so I'll go forward. Sorry. The third leg of ancestry is the genes. The whole genome, as I pointed out, it's a very large molecule. Those who are data scientists here may pay attention. How do you take a very large piece of data and then track it to compare and deal with comparison and all of that? And so how the scientists have gone about is look at what are known as SNPs. These are markers on a genome. Single nucleotide polymorphism, or STR, standard repeating tandem repeats that are present there. They are almost like fingerprints of the genome. So they track that, they take the genome, figure out all of these things, and then reduce it down to one matrix, and then take the same kind of information from another population and compare. That way they can figure out if one is derived from the other. It's a little too technical. There's a lot of mathematical methods involved in doing this type of analysis. Most researchers in this field do not do this, but some have done it, and fortunately, we have access to that data. And once you do that, you can then ask the question, is this population related to this population? For example, the Andamanese compared to people who live in South India. People who live in South India, are they related to the North Indian people? Those kinds of detailed genetic questions can be answered. That requires a lot of work, unfortunately. A whole genome analysis is required. And one of the major tools within that is known as principal component analysis, which is those of you who might remember, when you have many parameters involved, you try to discover the one that is more dominant and then try to pick the second one and then use that information to guide your comparison. So it's a mathematical technique that's now well accepted in the research field as a way to compare two different populations, if you will. Now, this field is less than 25 years old. For example, the early work in this area, people try to look at your blood group and see how your ancestry was. That's all thrown away. That has no meaning, in fact. And we really got to get to the first classification in 97, just about 25 years ago. And then the Y chromosome consortium was formed about a little over 20 years ago. And now in the 20s, major uptake was due to rapid means by which we can do genetic analysis. So it is the technology that has enabled us to get access to the information and the ancient genome analysis, if you will. There are many companies in the US that'll do a whole genome analysis for you for about $100. I'm not sure whether that's available in India yet. It probably is. Okay. Y chromosome and mitochondrial DNA, they're classified as A, B, C, D, etc. This is an overall map. This is the total map, if you will, as of 2013, because that was an easy access for me. But that's not as important as this one, which is I've customized that particular large, huge map to what is relevant for India. What is important for us is those that I've highlighted in red, for example, the haplogroup C, haplogroup C is in this category that we find in South India. And of course, haplogroup F, H, and of course, L. And approximate time when those haplogroups emerged, humans came into India approximately 70,000 years ago. Some would say it's 60,000. It's in the ballpark of about 60, 70,000 years ago. C was the first person to come into India. And then, of course, some of them stayed here, some of them marched on, and their distant cousins were discovered in Australia. So they went along the coastal route all the way to Australia, and that is now very well established. But 15% of the Australian Aborigines have the same Y haplogroup as C130, which was discovered near Turney by Professor Pitchempton's group. Now, these are pretty much native to India, and you'll see them in the South Indian population and also in North Indian population. The other mystery that is yet to be solved is R2. R2's origin is Central Asia, and there have been speculation as to how they came into India. Spencer Wells suggested that he came through Tibetan route into Gangetic Valley and then spread into India about 10, 20,000 years ago. We do not know. We don't have evidence for it. That's purely a speculation. But you find R2 right through India. We find a fair amount of them in South India. But their origin is very similar to R1. The R split into R1 and R2. This is what people have called as the Aryan gene or the steppe people, if you will. And they are related blood-wise, but they actually deviated from each other quite some time ago, 25,000 years ago. They're not the Vedic people. The Vedic people are the R1A. Now, two archaeological examples. This gentleman by name Virumandi was discovered by Professor Pichayappan some about close to 20 years ago, 15 years ago. He has a haplogroup CM130, and that's one of the earliest descendant of people who came out of Africa. They apparently came along the coastal route and then went up. And the amount of them that is present in India is very small. It's very shaded. This is, by the way, a mathematical model. So it's a very small number. There are many subclades of that, meaning derivatives of them. They're present in fair amount, not fair amount, it's in the orders of about 1 or 2% right through India. But you find a fair amount of them in Japan, in northern Mongolia. And there are some tribal groups in Japan who are actually distant cousins of this individual. And of course, they are dressed very differently, and they are literally related to him. This is one example to show how genetics can shed light on the early occupants of Theni district of near Madurai from nearly 70,000 years ago. This is for mitochondrial DNA. I'm not going to dwell on this very long. The key thing to remember is this is an old slide from 2015. All the L's are from Africa. The early ones that came into India was haplogroup M. Keep that in mind. And of course, a relatant is N. A derivative of that N is R, and derivative of that is U somewhere here. Now, India has got a lot of M's. If I were to pick among the women or the men in this group, if I were to pick one person, the probability that he is an M haplogroup is probably in the orders of 30%. The probability that it's R is another 30, 40%. So you're likely to be M or R, pretty much everybody in this room, very likely. But anyway, now that is the same whether you're a tribal group or you're a low caste person or a very high caste person, or just an ordinary farmer. It'll be nearly the same. Now, Professor Wright draws certain conclusions from that based on the idea that people who came into India were all men, not all men, mostly men, and women who came, because M is a very ancient haplogroup, and therefore they continue to propagate, if you will, by mating with the local women who are already here. Now, this is a more important slide. By the way, you'll be able to see this at this particular site. It's open access journal. The key thing to remember in this, the main thing that I want you to remember from this is M is present on in the entire slide. This is entire India, if you will. What are the various haplogroups of DNA, mitochondrial DNA that has been reported? The reds being the south, and of course, other colors are for different regions of India. The key thing to remember is M is omnipresent in Tamil Nadu and, of course, right through India. That is, the maternal lineage is almost universal. That tells you something about who we are as a subcontinent, right? Okay, now this is a part of the results of a 1,000 genome project. They took 1,000 genomes from all over the world and then organized it. I've just taken one small slice of it. The slice of it consists of Bengalis in Bangladesh and Gujaratis in Houston, Texas, in US, Indian Telugus in UK, Punjabis in Lahore, and Sri Lankan Tamils in UK. And the point that I want you to look at is here. All of those groups, the dominant population is M. And the next one is R, and of course U. These three pretty much covers all of the population of this broad class of Indians, if you will, which includes Bangladeshis and Pakistanis, if you will. And that will be the case if you were to take any population in India as well. Okay. Now, this is a study that was published by Anthropological Society of India a couple of years ago. So since M is an ancient mitochondrial DNA, people look for M1, which would be the first one. We have not discovered that so far in India, but M2 has been. Pretty much this community, this tribal community, Kuruman, 100% of them are M2, which is the most ancient mitochondrial DNA. And then Kurumbas have approximately 50 to 70%. And of course, Irulas about 5%. But so the ancient mitochondrial DNA is present in the tribal group. But if you go back and look at a high caste person or a low caste person, the numbers will not be terribly different. I'll share some of that results with you. Now, this is an example of how genetics has shed light on history. They had excavated a number of skeletons in ancient Mesopotamia, and they discovered 4 skeletons with a mitochondrial DNA of M49, M61, and M4B and B1. But in the area of Mesopotamia, there's nobody with that mitochondrial DNA. So the natural thing was it came from outside. And of course, there was a project that was launched to find out where they came from. So many genomes in India were examined by these authors, Vital, etc. And here is a summary of that. The M65A was discovered in Dindigul, and that age was 500 to 700 CE. And M49 was discovered 200 to 300 CE. And of course, M49 and M61 belongs to 3rd millennium BCE. That was discovered farther east over there. Where do I have that? I think that's further in the in Assam area. So you see residues of people from India in ancient Mesopotamia 4,000 years ago and also 1,000 years ago. There were 4 skeletons that were discovered. So here is a piece that supports the so-called Meluhans being people of Indian descent, if you will, right? Meluhans is a term that is you find in Sumerian inscriptions talking about Meluhans coming down from the hills for celebrating the building of a temple. And of course, Parpola has deciphered that to be of Indus Valley people. Indus Valley people, we could take that to be of India, if you will, right? So here is a case where genetic analysis provides information about history. Now, this is a pivotal, important paper, Raki Gargi skeleton extract, with the first author being Shinde from Pune. And of course, bulk of the genomic analysis was done in David Reich's lab. And they extracted a good chunk of the genome, not the entire genome, because it was the initial extraction was done at Lucknow. But so the analysis was done at Harvard University under a special agreement. The key information that points out is put here, the individual sequence, this is direct quote from his abstract, is a mixture of people related to ancient Iranians, hunter-gatherers, not farmers, because until this paper came out, the thought was the Iranian farmers came into Indus Valley and civilized the people in that area. This data disproves that. It says that was not the case. And there's another paper that was first author being Narasimhan, who also worked with, which shows the actual trajectory of the folks that came into Indus Valley. And they compared with what is known as Indus Valley Kline from this particular location in Zagros Mountains. Zagros Mountains is west of Indus Valley, probably a few hundred kilometers, 300-400 kilometers, and in a region known as Susa, where they spoke a language known as Elamite. The people who have theorized that Elamite has a linguistic connectivity to Dravidian languages, and also Elamite has been discovered to be the first script that has been written phonetically, just like how Tamil is written and how every Indian language is written. And so there's some connection, but there is no linguistic connectivity according to scholars. But that's yet to be explored, by the way. Now, the key result they found was this lady was really, they did a whole series of admixture studies. They compared her genome with many other people in the northern area, southern area, including Ethiopia, etc. And they discovered best match. Here is the mathematics telling you what to believe. The mathematics tells us she harbors genes close to Andamanese hunter-gatherers because they fortunately are isolated community in the Andaman Islands, and that has been wonderfully characterized by Dr. Tangaraj of CCMB Hyderabad, and that data was available. And the second part was that this is the lineage from hunter-gatherers from Iran, not farmers from Iran, because they were able to link to ancient genome data, which was about 10,000 years old. So this was a marked difference from the understanding of how India was populated. Major step forward in refining how people came into India, if you will. And of course, the authors speculate that the Indus Valley people invented farming on their own, not learned from the Iranian farmers. Now, that is, of course, a speculation. The reason is that data can be a little bit too ambitious, but he makes a cautious remark too. But something to keep at the back of mind. And the only way to answer these questions is ancient genome, for which India lacks that piece of data. Okay, how do Andamanese people look like? This is how they look like. These are pictures I took at the Tribal Research Centre in Ooty. It's just about 10 kilometers from the city, where they have a beautiful recollection of various different communities and tribals. And here is another picture of them. And this is how Indians probably looked in where we are about 70,000 years ago, very likely. And here is another one on the left. On the right is Nicobarese, which is the southernmost part of the islands. That's because of the backflow of haplogroup O, more of the Chinese type of folks, if you will. You'll find features more of that nature. They're also a tribal group, if you will. Now, if you look at Indus Valley itself, the faces, these are sculptures that are displayed in various museums. They don't look anything like any one of us. It has a very negroidal-like feature. And here is another person, a picture I took at Boston Museum. It's called as a spear thrower. So he has a jutting out of face, and that is the front part of the face is out. And of course, the famous ones are these. This is in the left-hand side is in the Delhi Museum. The right-hand side is from Kenoya's paper. And of course, the famous dancing lady, thick lips, broad nose, flat, and more flattish face rather than typical face of folks who you'll see in India, right? And you also see, this is also a sculpture from Indus Valley, such as this, more longish face, more alpine-like facial features. So they were quite varied. The speculation is it is a large civilization with varied kind of people, but of course some of them would have belonged to particular class, and they probably spoke more than one language. That'll be our guess. Now, the main point that I want to point out of Shinde's paper, which is with David Wright, are two things. One is the Iranian-related ancestry in South Asia split from Iranian plateau, and that occurred long time ago, as opposed to the conclusion that was prevalent before this paper came into place was the people from steppes came into Iran, crossed the Iranian plateau, and then came into Indus Valley, taught them farming, and they then was propagated further down. So that is not to be the case according to this paper. The second important piece of information that we get from this paper, and another paper that came out just a year before, is that the natural route of Indo-European language to have spread is from East Europe, like Poland, think about it, via Central Asia in the first half of the second millennium BCE. This is based on DNA data. It's not a guesswork. It's based on ancient genomic data and the chain of transmission now documented with the ancient DNA. And the travel path is published here. This is from Wiki, but it's inspired from David Reich's article. The people from Yamnia people went eastward, then turned around, and then came via Hindu Kush. They did go this way, but the people who came into India came via Hindu Kush into Indus Valley. This is an important distinction, an important change in the way people have written history. The ancient genomic data has provided us new information. You can dig this up all up in this particular paper. First author is Shinde, if you will. So the key thing is origin is pointed caps in steppes. Yamnia culture is what it is discussed as. They went through Central Asia. It's got names here. An interaction with BMAC, which is what I call, because they found traces of the steppe people's DNA in the ancient genomes of people who lived in this particular area in Central Asia. And then they arrived through Hindu Kush into India, into Indus Valley. Opposite to what the previous thought was via the Iranian Peninsula. Okay, I'm going to skip that. Second important piece of paper publication came out last year, about 6 months ago. These authors, it's available for you to download. They had information to generate where the haplogroup L. I'm going to focus a little bit on the haplogroup L because there's a lot of speculation prior to this publication that the Indus Valley people were haplogroup L. And we find haplogroup L in Tamil Nadu varying from a few percent to as high as about 20%, depending upon who did the study. And I told you that how the sampling method was not random, and so we don't have representative data. But L has been thought to be an important haplogroup of Indus Valley civilization. Now, the origin of haplogroup L is in this region, roughly, and they moved towards this region. So we see some residue. This is where, for example, where Susa is. This is Susa. And this is where the Zagros Mountains are. East of Zagros Mountains is where the IVC Kline is taken to be. I think we have 11 ancient genome data that has components of Andamanese DNA. And so we know that the Andamanese DNA people had some interaction with them on their pathway to Andaman. That's in David Reich's paper. And then they spread further into India. And of course, they're present in various different proportions in India. Now, these are mathematical models that predict the relative density by mathematicians. So I won't take this too seriously, but this is hard data. The data points are hard data. But what it seems to indicate is M27, which I'm going to focus a little bit on, and M20 are found in South India, as you'll notice here. And of course, that happens to be true to the extent of between 3 to 20%, depending upon which study you look at. And now here on the left-hand side are each dot on the left-hand side. This is years before. So this is 5,000 years before. So in the 5,000 years before, we have this many ancient DNA available, and we know what their haplogroup is, L. They all belong to a haplogroup L. It's a subdivision of M27. L, forget L1A1, but think about mutation 27 and mutation 76. They are daughters or sons of this lineage. And in 5,000 years ago, we have 1, 2, over there 3 and all of that. We have at least 3 pieces of data, but more than 2,000 years ago, we have a bunch of data. And what the authors did also went sampled people from current day population and have just expanded the red part of it that's out here, and you'll find them to be people from Balochistan, people from Sri Lankan Tamils, Singapore Tamilians, and Telugus, and of course, Gujaratis as well, because they're near Indus Valley, and of course, various different regions, Telugu, Sri Lankan Tamils, and Gujaratis, a fair number of Gujaratis. So what this says is this individual over here is actually a descendant of this particular individual who's a haplogroup of this lineage, if you will. Now, the most important thing from this slide, first time it came into existence just a few months ago, or 6 months ago, is the connectivity of large number of reds, which is primarily Indians, South Indians, if you will, compared to the blues, which are more in Kuwait and Europe, more in Arabia, etc. This particular sublineage of M27 were the lineage that pretty much populated not entire South India, but a fair number of them came into India. So this is an important paper that provides us this rich information on movement of people. And this was possible only because we had access to ancient genomic data. Otherwise, you're guessing stories, right? You're making up stories. Now, this is hard evidence. Now, another important piece of data published about 6-7 years ago was this individual was interested in the Jat population of Rajasthan. Remember, Rajasthan is at the edge of Indus Valley. And in fact, there are 3 major late Harappan sites in Rajasthan. In fact, I almost tried to visit one of them, but I couldn't during my recent visit. So 1/3 of the Jat community, 37%, according to this author, belong to haplogroup L. They didn't do the refinement of subdivision. So these individuals and the L27 that I mentioned, people who spoke Sri Lankan Tamils and Telugus, they spoke the same language probably some 4,000-5,000 years ago. There's no difference. Key point to remember, although the Rajasthani here, Jat community, speaks an Indo-Aryan language, they probably spoke an entirely different language, probably Proto-Dravidian language, some 4,000 years ago. So language and your genes have no correlation. The language you speak is pretty much where you have lived you and your family has lived the last few hundred years. And many of you might have been migrants into Tamil Nadu or were original inhabitants. That connectivity is not quite there. And that is sometimes it was difficult to convince people at the last Zoom meeting, but that is an important message I want you to carry forward. The language you speak is based upon your history, if you will, rather than your. Now, this is an important piece of paper Professor Pitchapin and Spencer Wells did. They sampled primarily communities in this region, tribal groups substantially, and different castes, etc. I don't like this study based on caste. It sort of divides us immediately. I'm going to go through this somewhat quickly. And he looked at what their Y haplogroup only. He didn't look at mitochondrial DNA. And the key point that I want to notice is the dominant ones, although you can look at it at your leisure, the key point that I want to point out is the dominant ones in this travel group and the various communities was haplogroup F, which is derived from C. And nobody knows exactly where it started because we don't have ancient DNA data to support it. But we know it was derived from C M130. Guru Mandi's relative actually gave rise to this particular community. And H1, this is another haplogroup that is completely found in India and also derived from the original inhabitants who came into India 60, 70,000 years ago. And of course, KM9, which is also a similar kind of a haplogroup. So And of course, you also see among them people with R1A, oh, I can hardly see this, at levels of 15%, 7%, 8, 9%, etc., 10%. And these are the people who came via steps through Hindu Kush and then into India, right? So they're also a blended society. The Adivasis, the tribal groups are also a blended society. The key point that I want to make in this particular slide is the R is associated with Central Asia, far away, and it's split into R1 and R2. The R2 folks, we have no solid, direct, ancient genomic data to substantiate it. Spencer Wells has speculated that it was these folks, R2 from Central Asia, came via Tibet into the Gangetic Valley and then spread into India about 10,000 years ago. Again, that is a speculation not based on hard data. On the other hand, R1, the other division, R1A and R1B, we have solid ancient genomic evidence. We know how R1A traveled through into Hindu Kush. We also know how R1B, we primarily populated Europe. We also know that because we have a lot of ancient genomic data. So this part, hopefully, somebody in India can fill this up. And I'm sure this result is there somewhere in ancient genomic data of some Indian skeletons that is yet to be discovered, if you will. Now, the final part of this is summarizing Pichapin's results. Out of 1,600 samples, L was less than 3%, which is the LM27 I mentioned. C was less than 0.3%. And the dominant ones were F, H, R, and R2. That is what his conclusion was. And of course, R2 it appears, all scholars say it is native to India. You don't find R2 anywhere else, although they came from Central Asia. They came into India long before R1A people came into India, the so-called Vedic people came into India. It's a much more ancient migration into India. Again, that is to be substantiated with data. That is the current speculation among scholars. Now, the next few slides I can show you quickly. The key point is this set of authors looked at, again, Y-haplogroup of various different communities. And what they discovered was, I've summarized the main result here. The results are really from here. C was 3.4%. Remember, it was 0.3% in Pichapins. That's because the way sampled was not exactly identical. They were not randomized samples, but they actually, I think I would call that as a biased sampling. Maybe they just took whoever came to their door to take samples from. And F was 10%, L was 13%. Remember, it was only 3% in the earlier study. And H was 20%, R1A1 26%, and of course R2 is 10.6%. So that's simply summarizing this particular slide. And they did a mitochondrial DNA of the various population in South India, primarily from Tamil Nadu. And the dominant portion is M. M dominates. So I think M averages total is about 62%. And there are others which are present. U is present also in this population. And I must note that U2, the lady that was discovered at Rakhi Gargi, was of haplogroup U2B. And so there are relatives of her living in Tamil Nadu, if you will. Okay, I picked this example, probably one of my last examples, is from a tribal population study in Gujarat. And the name of the tribals are listed here. And the reason why I picked it up was this pretty much shunts with the Indus Valley civilization. There are Indus Valley sites in Gujarat. And their Y haplogroups are R1A is 53 in this population. I've summarized it here. Pre-2000 BC, 54%, 19% post-2000 BC. That is, they blended, the tribals blended with the incoming Vedic chanting people, and of course have created their own lineage, if you will. So it's also a blended culture of ancient folks who came into Indus Valley and their own native population, if you will. So the message from these slides are tribal communities or various different communities exhibit fair amount of various ancestry, not a single ancestry in terms of Y haplogroup. When you look at mitochondrial DNA, they predominantly belong to the same population group, M or R, primarily, and a small amount of U. Those are the main messages to get out of these slides. Okay, now I go to a story, to my own DNA story. The reason why I included this was to validate the method that the methods that people have used to illustrate that there is some validity to some of these arguments made by scholars. So 23andMe, my daughter-in-law was very active in this, so she took samples from my son, etc. And of course, 23andMe does not know my son's sample from mine, and of course, her father's, and so on and so forth. So it displays Raj Mutharasan as the close. It identifies him as my son because he harbors 47%, somewhere here, 47% of the genomic data as I have. He's supposed to get 50%, but that's an approximate number. And so genomic mathematical models can predict relationships very accurately. And then I discovered that this individual, Karasu, and so on, they came from the same village as my grandmother, if I remember right, comes from, not very far from Kurjibadi. And they migrated to Singapore some 70, 80 years ago. Their daughter went to Australia and she married an Australian. And of course, that Australian, I was in touch with email exchange, and we have had exchange of information on where they came from, etc. It so turns out they're very close relative, but I don't know how they're related. The reason is they say 3 and 5 segments. That means they're very, very blood related. I mean, they're within few cousins, if you will. But I do not know. I haven't tried to sort this out because the older generation is gone. The younger generation, their granddaughter is doing nursing in Tasmania. I've been in touch with her too. But of course, that population group really comes from the same village as my grandmother comes from. And then, of course, Sumia Khan is supposedly close. And of course, Patel, a Gujarati, as the name would suggest, is a close relative. Now, the key thing to remember is they compare. It took me some time for 23andMe for them to share with me how do they figure out this segment. This is a unit of DNA distance that is identical between the individual and mine. And 6 means it's approximately 6 million base pairs. So it's a fairly long segment. It is not a whole series of segments, but it's significant enough to say you're related. And of course, if they made that number 6,3, I'll have millions of relatives. And of course, they don't want to deal with large data. If they make it too large, I won't have any relatives. And so their business model will fail. So this is really optimized for their business model. But they also allowed me to download my entire genomic data, which I have on my computer, and then I can submit to different databases to do detailed analysis if I want, for example, to look at admixture of components. That way I can discover other DNA relatives if I so wanted. But anyway, the key thing to remember is I have another Patel, and this lady is from Karachi, and I think that might be my last slide. Yeah. I have, as far as from this particular collection, I have 2 relatives who are in Karachi. They're not very distant. They're within about a few thousand years. And 2 from Gujarat, 1 from Maharashtra, 1 from Hyderabad, several from Tamil Nadu, and several Fiji, Trinidad, and Vietnam. Fiji and Trinidad are laborers who went from South Arcot district when the British took them for sugar plantation. And of course, they remember having come from Cuddalore, but they cannot place the location of the village, etc. So I was in communication with them. They know they came from Cuddalore, but they do not know any other relation. They're about 3, 4 generations down after they migrated out, particularly the Fiji. So the fact that my ancestors or relatives trajectories from Indus region. I'm taking Karachi as an Indus region. to Tamil Nadu via Gujarat and Maharashtra seem to suggest to support Balakrishnan Ayya's hypothesis of Indus to Vaihai trajectory, right? Journey of a civilization. So I'll stop there to say the final summary is folks in India are a blended population. There's nobody called pure this or pure that. One may be more of one and less of another, but never the same. There is no connection between, I would call, say, if you're the possible definition of Tamil ancestry is non R1A ancestry. And that also is questionable because it's not a precise statement from a scientific point of view. The mother tongue and genetics hold no correlation at all. It's possible you were speaking some other language 300 years ago. And so I'll stop there and answer any questions you may have.